So good afternoon, everyone, and welcome to the Region 7 Science Program's webinar series. My name is Eleodora Chamberlain, and I'm the Region 7 Science Liaison. The Regional Science Program is pleased to host a research rock star among rock stars, Laura Webb, with the Field Services Branch in the Region 7 LSASD Division, that's the Laboratory Services and Applied Sciences Division. Laura began her career at EPA Region 7 as a student intern in 1988, while earning her degree in chemistry from UMKC. Upon graduation, she was hired full-time in the regional laboratory, and during her 16 years of analytical work, she specialized in organic sample analysis and became the regional expert in the analysis of dioxins and furans, even analyzing samples from the World Trade Center after the terrorist attack. In 2007, Laura was transferred to the water monitoring team, where she conducts field assessments and laboratory analyses for regional and national programs. She also maintains and operates the region's mobile monitoring lab, which is an accredited bacterial analysis lab. Her current assignments range from water quality monitoring to national assessments to E. coli analysis for emergency response in the mobile lab, and is a regional lead on cyanobacterial monitoring and analysis. Today, she is presenting on her RARE, which is a regional applied research effort research project on harmful algal blooms in urban environments. This is a live webinar and it's being recorded as we speak. If you have any questions for Laura during the webinar, please write them in the chat and we'll address them at the end of the presentation. Please put your mics on mute and we will open up the lines later for questions. So at this time, I am going to stop sharing my screen and hand it over to Laura.
Thank you, Eleodora, for that introduction. Let me get my screen shared here. Just can tell me if that's working. Looks like it's red on my end.
It looks good.
Okay, great. Well, let's get started. Again, thanks for the invitation, Eleodora, and thanks everyone for coming to watch the presentation. Up front, I want to do a confession that I'm a chemist, not a microbiologist. So if I stumble, that's why. This is some of this stuff is a little over my head. I've had the help of our microbiologist, Meredith Mattis, to kind of explain a couple of things to me in words that I can use to explain them to you. So that's cool. This is work based on a RARE project that we did in 2021. My collaborators were Marcy Tidd from Region 8, Heath Mash from ORD, and Dr. Jingreng Liu, also from ORD. And the paper that has some of this work presented was put together by Dr. Liu's lab in ORD, and the reference is there on that title slide, as well as a link to the paper that they put together. We also couldn't have done this project without the regional labs, so big shout out to them. They did an incredible lift to get us some of this data, and the sampling teams in all the cities, especially to my sampling partner, Regina Klepico. Okay, so let's get started. I'm going to talk about what are cyanobacteria and why do we care. Some of you may be new to this field. Some of this may be repeat, so just bear with me. I'm going to describe the RARE project, look at some big picture data, describe some of the techniques we used in the project and in the paper, and then some specifics that the paper talked about as far as the toxins and the genetic applications. So what are cyanobacteria? I kind of like to break this word in half, right? Cyano, that blue-green pigment, or phycocyanin, it is a pigment along with chlorophyll that these organisms have. Chlorophyll, yes, it does photosynthesize, right? So it uses sunlight to produce their own food with this chlorophyll molecule. Kind of makes them sound like a plant, and they're commonly referred to as blue-green algae, but they are actually bacteria, microscopic organisms, not plants. They do need nutrients to grow, just like other plants or other organisms, really. They're found in freshwater, estuarine, marine waters, and the excessive growth can lead to blooms. That growth can be triggered by different nutrient parameters, just the competition between the organisms that are in the water body, and other triggers that can then make these cyanobacteria grow uncontrollably. And unfortunately, some of these produce toxic compounds, and those toxins are what we're going to talk about today. Especially pets are real susceptible to the toxins in water because pets go all in, right? You know, they get in the water body and then they lick their fur when they get out and they chomp on things in the water. So that's why we've seen an uptick, I think, in dog deaths related to cyanobacteria. But there are also human health concerns and economic concerns. I know that in recreational water bodies, when you have a bloom, you have to shut some stuff down. And so towns around that water body, people that are reliant on that recreational influx of people can be impacted by these blooms. Okay, so let's keep going. So cyanotoxins, this is the organisms that can be produced by these cyanobacteria. This paper in particular deals with saxitoxin, and it's also referred to as paralytic shellfish poisoning toxins. They're produced mainly from marine dinoflagellates. Gosh, I can't say that word. And by freshwater cyanobacteria. They're neurotoxic, so potential symptoms range from death, numbness, tingling, to death from respiratory failure. Exposure to the marine saxitoxins are usually through ingestion of contaminated shellfish. But then in freshwater, it's generally a dermal exposure route. We also have several other classes of compounds that are toxins that are produced by cyanobacteria. There's over 200 different forms of microcystin, and it's a hepatotoxin, also affecting the kidney and reproduction. Nodularin is also a hepatotoxin. It's been known to bioaccumulate. Anatoxin is a neurotoxin, serious neurological distress with signs ranging from loss of coordination to death due to respiratory failure again. And this is the one that pets seem to be affected by the most, mostly from benthic cyanobacterial blooms. And, you know, that's not what we're talking about here, but we do have some anatoxin in these lakes as well. Cylindro is a liver kidney health effects. It's also been known to bioaccumulate in aquatic and invertebrates and vertebrates. EPA has developed drinking water criteria and recreational advisory levels for microcystin and cylindrosmosin. And they're also working on anatoxin and saxitoxins. But some of the states have gone and done their own work on this and proposed their own limits on all of these toxins. The study itself, kind of a little bit of background on the study. We looked at urban lakes in 3 different cities. We started in Denver, Colorado, Kansas City, and then Cincinnati, Ohio, where our ORD lab is located. We did several different types of sampling. One is called SPAT, and that refers to a technique called solid phase absorption toxin tracking. It's a mesh bag with a sorbent inside, and you absorb the toxins for a week-long deployment. At least in this study, it was a week-long deployment. Techniques is called passive sampling, so it gives you a look at the toxins over a long period of time as opposed to a single grab sample. And then the other things that we did some water grab samples. We had 8 deployments here in Kansas City, 10 in Denver, and 14 in Cincinnati. And you see my partner in crime there, Regina Klepico. Yeah, that was our April sampling, and it was snowing. So we also have some pictures of Denver with them working in the snow. So interesting times. We analyzed the grab samples for multiple parameters, over 200 different parameters on each sample. And again, we couldn't have done it without the labs, the regional labs here in Kansas City and in Denver, and then the ORD lab in Cincinnati. Here's a couple big picture slides. Chlorophyll is that pigment, right, that produces or that you use for photosynthesis. Chlorophyll is one of our basic measurements, and it's used to estimate production in the system, or how much biomass, how much is stuff growing. And you can see that Denver and Kansas City really outproduced the lakes in Cincinnati. We had more stuff growing in our lakes. In addition, Kansas City had way more cell counts than either of the other 2 cities. And these cell counts are an estimate of the cell counts. So we definitely outperformed here in Kansas City. And then fluorometric analysis of pigments, especially this phycocyan and chlorophyll ratio, is an excellent indicator of cyanobacterial dominance. And you can see that Kansas City lakes definitely were outside the norm, right? And we predicted, or we could see in the samples, that we did have a lot of cyanobacterial blooms. Here's a look at our sampling locations here in Kansas City. A better look is this land cover data map. You can see that the dark reds are very highly developed areas. The pinker are the more less developed, but still urban areas. We're looking at a lot of impervious cover with these red areas. Our reference lake is Lake of the Woods. You can see that it's got a lot of forest cover in it and a lot of open spaces. Those are probably parks, recreational areas associated with it. So a wide range of lakes there, very small watersheds. Lake of the Woods is probably our biggest watershed, and it actually includes some of the zoo. So it'll be interesting to do some more study on that watershed in particular. Another big picture slide from, these are just some nutrients that we found here in our Kansas City lakes. Of note, Lake of the Woods is really low in nutrients. That's good. That's our reference lake. That's good to know. The three that had significant phosphorus concentrations, South Lake, Shamir, and Big 11, well, that's where we had the blooms. So phosphorus is definitely a strong indicator. It's one of those things that are associated with almost every harmful algal bloom. All right, so I'm going to talk about a couple of the techniques now. Flow cytometry, or FlowCam, is the instrument that we use. We're looking at cyanobacterial abundance, and we can look at taxonomy in a very high level taxonomy, to the maybe genre level. You need a microscope really to get more specific to the species. You inject that liquid sample and it flows through a cell with an imaging system. There's a laser that that triggers the cells to fluoresce. Those two pigments again, chlorophyll and phycocyanin, fluoresce. So you shine the laser, it fluoresces, the detector notices it, and it says, take a picture. And so it snaps a picture. And these images you can see on the screen are individual pictures of particles that have gone through that flow cell. You can get hundreds of images per sample. We usually run about 1 ml of sample at a time. The software is designed to take 40 physical attributes like size, shape, color, circularity, those kinds of things, and bin these according to these shapes and the sizes of these different particles. It's very fast, but it's not very specific, especially when it comes to straight chain cyanobacteria. So all of these pictures there on the left in that collage of images, those are all Dolichospermum. Well, do any of those look alike? No. That's why it's so hard for this particular technique to bin these by similarities, because each of these pictures is unique. So what it's trying to do is build a box of shapes, or of attributes of these shapes, and then segregate things into those different bins. It's very difficult for things like Dolichospermum because each image is unique. The straight chains there, you can see some of those in that picture up to the right. Well, those are a little easier. However, some of these look exactly alike. You know, you're not getting a really good picture on the FlowCam, so you can really separate what any of these strands look like. For instance, that strand that's third one on the bottom row from the right, is that the same as the two strands on either side of it? Probably not, but it's gonna, the software is gonna bin them all into one thing, right? So you have to go in and pull these out with just your eyesight. So it's kind of time consuming. You can get a really great overview of what's in your sample, but to actually do taxonomy on this instrument was really, really hard. So I will present some FlowCam data, but you'll see I've clumped all these strands into one bin because I can't tell them apart. Okay, so moving on from the taxonomy, right? We're looking at toxin analysis. two techniques that we used, enzyme-linked aminosorbent assay and liquid chromatography mass spectrometry. The advantage of ELISA is it's really inexpensive. It's relatively easy to do, especially when you have automated systems that we have now. It does give one value for the total toxin. For instance, it says all the microsystems are all clubbed together, and it gives you one number. You can get quick turnaround on these, but you have to run each toxin individually. So it kind of compounds the time that you have to spend on it. We can, I don't know who's not muted, but it would be great if you muted yourself. And we can do it portable. We can do it in the mobile lab. We can throw our little plate reader into the lab and take that in the field with us. LCMS, it's way more expensive. It's way more complicated. It requires the lab infrastructure. However, you can look at individual congeners for things like microcystin. We can look at not all 200 congeners, but we have standards for 15, 20 of these, right? So we can look at each of those individual numbers. And it can do multiple toxins in one analysis. However, right now we don't have a method for saxitoxin on LCMS. Here's an example of that congener data. So this actually is what prompted this study. I took some of these samples with me when I did an R2P2 in Cincinnati with Dr. Mash, and he ran them and he found some patterns that he was not expecting. He was not used to seeing patterns like this with his reservoir samples. So that's really what started our deep dive into these urban lakes. So we're seeing, I just have what, 3, 6, 9, 10, 11 congeners here that were detected in these samples. And there's more, but just depends on how many you run at a time. And you see you did different congener patterns, right? So massive LR with some LA in the top graph, but kind of equal contributions of RR and LR in the Shamir Lake, and then totally different in the Denver Lake. So this is what we were investigating, is why these patterns were different. You're all seeing microcystin. The ELISA is going to give you one number, but it's not one number. It's a summation of all these numbers. So that's what started us looking at this project to begin with. All right, so now we're going to get into the genetic techniques. There's 2 slides here, and I got to thank Meredith Mattis for helping me put these together, actually putting them together for me so that I could explain it. So I'm going to show you a chart from the paper that talks about this metagenomic data. What it really is it's very useful to get the species, right? So we're going to extract the DNA from the cells, and we're going to break it apart, and we're going to compare the DNA sequences to a library of DNA that we have and match up to the species. So we can get genus and species present in the samples, which is really cool. And it's way expensive. It's not very widely available at the regional lab. That's why we do things with the FlowCam and the microscopes, right? Because that's the way we have to do them. But this data proved me wrong in some of my identifications. So it's very useful. It's very cool. One of the techniques that we're really developing, right, to use for cyanobacteria. And then the other technique is qPCR. So this is polymerase chain reaction, right? So you're looking at DNA sequencing and breaking it apart and checking out what types of genes are in there. You got to know what you're looking for, right? So 16S is the phylum-specific marker. It identifies the presence of cyanobacteria. And then this SXTA gene is the gene that can code for the production of saxitoxin. So, but remember, doesn't indicate the bloom is toxic or that the species is toxic. It just indicates that the gene sequence required to build that toxin is present in the cell. So you can compare different water bodies and see, which one, based on the total cyanobacteria that's present, how much of the toxin gene is there, or how little of the toxin gene is there. So as you see here, Meredith gave this example, the potential bloom toxicity of bloom A is greater than bloom B because you have more of the saxitoxin gene in the whole set of genes that you're looking for. We've also looked with ORD looking at RNA and not just DNA, but the RNA, and that's the stuff that's turned on, right? It's actually actively producing toxin. So we've done some looking at how quickly we can see that in a system. Generally, we can see that before we see the toxins. So it's kind of a, we can look to predict, right, how toxic it's going to come, because we can see this first, this qPCR for the DNA, but we can also see it for the RNA. And if you're interested in this, Dr. Jingrang Liu is going to do a presentation tomorrow as part of the HABs and hypoxia series on this predictive ability of the qPCR work. All right. So here's a look at Big 11. I'm going to focus on this lake because the paper does. We're going to talk about a couple other lakes too, but this is the big one that I have data to show for. It's a very urbanized area. That's basically what I'm showing you with this slide. It's highly urbanized, a lot of impervious surface, and it's just down the street from the lab. Flow cam images. It's very green. When we first went out there in July, we already had this massive bloom that was covering a good portion of the lake. These are the flow cam images on the left and right. On the bottom, that's microcystis on the left, and that's a filament on the right. I thought it was something different than what the DNA is going to show you that it is. So, but it's definitely a cyanobacteria, and it's definitely a filament. So that's how I'm bending them at this point. So we really had a high abundance of microcystis for sure at the beginning. Some interesting indicators that may have happened during this lake. When we went out there, we had a very high concentration of phosphorus, way higher than the other lakes in Kansas City. And this phosphorus caused growth, right? It was the food that fed the organisms. You have a very high chlorophyll concentration, which means it's a very productive lake. There's a lot of stuff growing in there. And the PC chlorophyll ratio was high, indicating it's being dominated by cyanobacteria, which I can see by looking at the water. Okay. We don't know if it's harmful at this point, right? Because all we're looking is to see that we have a bunch of cyanobacteria. We don't know if it's harmful yet. Okay, but then we looked at the toxins, right? So you've got the LC-MS-MS graph there on the upper left, and then on the graph on the right, the straight line there is the ELISA concentration by, or the microcystin concentration by ELISA. And you can see we had a lot, we had a ton of microcystin, 243 micrograms per liter was the highest concentration there, and that's a lot. We did have good correlation between the highs on the LC-MS and the highs on the ELISA. That's always nice. And then the graph on the right shows the flow cam particle count. So this is the number of organisms that I counted for each type. Early dominance was microcystis, which you saw the picture just like I did. A gradual move towards Planktothrix. And I identified it as a Phanezomenon, not Planktothrix, so the DNA helped me fix that. But that was the shift, the species shift there after the phosphorus went away, right? We shifted species. But of note is we also had anatoxin and saxitoxin in this lake. The saxitoxin was the highest we found in the study. So it's a mess out there. Not something you want your dog playing around in. The thing about the anatoxin and saxitoxin, we don't have limits for them EPA-wise, right? But Oregon has gone so far as to develop advisory levels for dogs because they're super sensitive to these toxins. And the level for microcystin is 0.2 micrograms per liter, anatoxin is 0.4, and saxitoxin is 0.02. So as you can see, we clearly exceeded that limit for saxitoxin. Way over the threshold. And I'm surprised we don't see more dog deaths in the urban core from these toxins, but maybe we're not looking for them either. Okay, keep going. I'm looking at the saxitoxin concentration is the purple line, and on the right, the saxitoxin gene abundance is in that yellowish brown line. And then it's backset by the flow cam observations. So you can see the genetics link the saxitoxin to this early abundance of this cyanobacterial filament, which I'll show you in a minute, was a phanosomenon. Microcystin was probably produced by the Microcystis cyano, this line here. And then you can see the genes also, they dropped way off after that phosphorus was used up, and the species shifted to Planktothrix. So this is a table or a chart directly from the paper, and this is their, we talked about the DNA sequencing that was done, the metagenomics, right? So this is their results from the metagenomics. You see that in this lake, early on, we had an abundance of a phanosomenon, and then it shifted. I think it followed that phosphorus line. It shifted to where we had a little Raphidiopsis, but it mostly was dominated by Planktothrix there at the end. And so that's what we have with the metagenomics. Let's take a look at this blue area under the curve is the ELISA concentration of saxitoxin, and the orange line is the genetics, the genetic marker for saxitoxin production. And you can see that started off high and it dropped off. The concentrations dropped off pretty dramatically, and this is a log-based scale for the genetics, and it dropped off as well as the season progressed, as the species shifted. Just to look at again, this is the flow cam data. You can see that on the 7-6, how the species started to shift there. Early Microcystis domination with some phanosomenon, and then it shifted all to Planktothrix in the end. This is a heat map also from the paper directly. And if you look at these two cornered tabs here, Big 11 Lake correlated very well the saxitoxin concentration and the genetic concentration. Both were attributed to a phanosomenon. And it was not the most abundant species, but it was there enough to produce that saxitoxin in the early samples, and then dropped off later on. It was on the KDHE HAB advisory for 21 weeks that year, and it still exceeded the cell count limit even in October. And also you can see on the bottom of this table, you see South Lake. That's where we're going next. South Lake was also on the HABs advisory for KDHE. We also had 3 different toxins at South Lake: microcystin, anatoxin, and saxitoxin. And anatoxin was the big winner here. It definitely was higher here than it was at any of the other lakes. It's a really great example of how the toxins tracked with the genetics. So you see the dark blue line is the concentration from ELISA, the light blue is the LCMS concentration, and then that green line is the anatoxin genetic marker. And you can see those all were up when they were all up, and they were all down together. It's a really good picture of how this data corresponds with each other. And had we been out there earlier, we might have seen that genetic signal before we saw the toxin signal. We've definitely seen that in the past. This is a slide of the flow cam images from South Lake. Early on, it was a lot of green algae, a lot of diatoms. It really wasn't overwhelmed by the cyanobacteria. But as the season progressed, we gradually lost almost all of that green algae, and it was dominated by cyanobacteria. Just to look at that genetics data from the paper, you see they attribute almost all of this to that yellow there is Planktothrix. So they've got a really big Planktothrix bloom going on there. And I'm not sure why the genetics data doesn't really pick up microcystins very well. They also are showing some Raphidiopsis, which I, again, lumped into that cyanofilament category because I can't tell them apart on the flow cam. And if you see on 8.3, we did have a pop-up of dolichospermum. We saw that on the flow cam and we saw it on the genetics. So that's kind of a nice confirmation, at least in my mind, that we are seeing everything that the genetics sees in the lake. Okay, moving on to one more lake, Shamir. This is, especially with a flow cam, taxonomy is really difficult. I've tried to color these similarly so that you can see purple on the big graph there is microcystis. You can see the purple line on my graph. They didn't pick up very much microcystis, whereas it was the dominant taxa for many weeks on the flow cam. But we also saw a lot of dolichospermum, and that is being picked up on the graph. With the greenish colors, right? The interesting thing about Shamir is we had two different, what I think were probably two different species of dolichospermum, and I'll show you that picture here in a minute. But if you look at the overall abundance and how mine go up and theirs go up, it tracks really well. So that was gratifying, right? And then the brown line here on this chart is the 16S cyano. So that's giving you a general abundance of cyanobacteria from the genetic side. The green line is the actual microcystin gene production gene, microcystin production gene, and it tracked really well with the dolichospermum. So that is probably what caused the microcystin in this lake. If you look at this is just looking at the total microcystin by ELISA and the genetic marker for microcystin. They tracked excellent, so that's cool. Then you put that picture up again of what they found genetically on the metagenomics, and it was dolichospermum that was causing a lot of that growth, it looks like. The three big high areas of dolichospermum were where we saw the most microcystin. I don't know why I had so many clicks on that graph. So here's a look at the flow cam image there on the left is the dolichospermum that started out as the dominant taxa there. And this is the LCMS trace here of the two of the congeners that were dominant here in this lake. We also had anatoxin and saxitoxin, but they were much lower concentrations. On the right is the flow cam readings, the images counts, the image counts. And you can see that dolichospermum is the primary here, but I also had microcystis in this lake, and it didn't look like the genetics really picked that up very well. About mid-season, we shifted to this big fat dolly, is what I call it. It looks to me like a different species, and it looks on the profile, the congener profile, it looks a little different there in the August profile versus the profile at the very beginning. And we also had the microcystin marker there, that dark green line. It traced with the dolichospermum. Okay, so we're looking at saxitoxin here in Shamir because that's what the paper focused on. You do see positive genetic hit as the toxin concentration increases. So again, it's very useful tool for prediction. And this is part of Dr. Liu's talk tomorrow, where he can use these genetic markers to predict. Just a quick look at the heat map for this particular lake. It shows saxitoxin is attributable to Aphanizomenon, microcystis, and Planktothrix. So that's kind of, or at least it correlates with that. So that was kind of interesting. But this lake, definitely dolichospermum was the dominant taxa, and there is no correlation with saxitoxin, so probably not dolichospermum that's producing that toxin in this particular lake. I've shown you this picture before, but just to look, Big 11, we decided it's probably Aphanizomenon producing microcystin. In Shamir, it's dolichospermum, so they're different patterns, right? And that's what we really wanted to dig out. And then the Denver Lake has a different pattern entirely. So it'd be interesting to go in with that heat map concept that they did in the paper and do that with microcystin. So I think that's kind of one of the next steps that we're going to take. Just to wrap up, again, it's a chemist's interpretation of molecular data, genetics data. So forgive me if I've totally not explained it very well. Read the paper. It's really fascinating. I thought I've read it like 3 or 4 times, just digging in. This is like the third or fourth time I presented on the study, and I know Marcy and Heath have both presented, and each time it's a little different twist, right, on what's going on. So there's a lot of data left to dig into, a lot of things still to learn. Genetics are a valuable tool, but so is the microscopy, the flow cam, the ELISA. There's a lot of tools that we can put together to look at this. And again, the paper goes much deeper, so take a look if you're interested in. And that is all I have. So I'm going to quit sharing my slide.
Thank you, Laura. So let's see here. Let's go. We have some questions. Let me open up the chat. The first one, is the South Lake site the South Lake Park in Overland Park, Kansas, near 80th and Marty Streets?
Yes, it is.
Thank you. Another question is that site, I'm not sure which site there this person's referencing, has always had high algae counts. So Mark, if you want to be specific on what site you're referring to, feel free to open up your line and ask the question or your comment.
Yeah, that lake is familiar to me because I walk by it every time I walk to high school there at Shawnee Mission West because I walk by it a lot and it always had lots of algae in it. And we never let our dogs play in it because it was always super green, you know. I just wondered if it was the same lake.
Yep, that's it. And I wouldn't let your dogs play in it for sure.
Thank you. Next question. Lake of the Woods have very high algal blooms. Did you say that that lake is your base lake, correct?
Yep. So the Lake of the Woods always looks super green, right? But it's not cyanobacteria, it's duckweed. And it floats on the surface, and it's not even an algae, really. It's an aquatic plant. We've gone out there on multiple occasions and had to wade or paddle through this huge, massive covering of duckweed on that lake. But it is not a harmful algal bloom. It is not toxin producing. At least I've never seen toxins in that lake.
Thank you. Let's see, there's some love being shared on the R2P2 program. That's the Regional Research Partnership program with ORD. That's coming up soon, and you'll hear about that from me later on in the year. And then we have your partners in crime. So we have Regina Klepakow and also Meredith Matz chiming in here. Next question or comment. Please consider St. Louis, Missouri urban lakes in mind for potential future effort sites. Many of those water bodies are prone to HABs. Is this something you might be considering?
Yeah, it's a great observation. We've always wanted to take this urban lakes project and push it out into other urban areas. We're going to do some of that with some environmental justice work we're doing this year into a couple of those different communities. So we'll certainly, I've always thought St. Louis would be a good option for this because it's only a few hours away right from the lab. So we'll definitely keep it in mind, and maybe I can reach out to you and if we can figure out what lakes would be best to study. We'll see if management can go along with that. I'm always into doing urban lakes, but my managers might not be.
Maybe we can find a research project on that to expand that.
There you go.
Let's see here. Mark Lesher has a few comments. Don't let your dog swim, play, or drink in these lakes. Isn't that right?
That is totally true.
Mark also says we use more fertilizers than runoff into these lakes. Okay. Another comment, why is Kansas City an overachiever in comparison to Cincinnati and Denver?
Yeah, and it might have just been this year, this particular year that we studied. All the lakes that we studied were known bloom lakes, right? So it might have just been the combination of we were warmer or we had more sunlight, they had less. It's just always hit and miss. Seems like every time you throw money at a project, that's when it doesn't bloom. We had that happen out at Milford a few years ago. But it's just a combination of things between the sunlight and the heat and other stuff that's going on in the lake, because the cyanobacteria are competing, right, with all the other organisms in the lake, and it just happens to overcompete. It happens to win that battle in these particular lakes.
Thank you. Let's see. How would drought or excess rain affect these results?
Yeah, totally going to increase the number of blooms and the severity of blooms, the less water that we get into the systems. And if the temperatures keep rising, right? So climate change is definitely affecting harmful algal blooms. It may even be affecting which species are dominating in these lakes and which toxins are being produced or how much toxin is being produced. We don't know all the answers there, but I would say for sure you're going to see a correlation with climate change and increase in these harmful algal bloom events.
Thank you for that. Let's see here. Is there a method of treatment, water or wastewater treatment, for the reduction of phosphorus?
So for these urban lakes, you'll have to go back and think about where the phosphorus may be coming from. That big phosphorus spike in Big 11, the only thing I can think of is somebody over fertilized and then we had a rain event, right? And it washed it all into this little catchment. And that's what a lot of these lakes were built for, right? Is just to catch runoff and as a storm control, right, systems. So yeah, I would say the biggest thing for these urban lakes, at least the ones that we look at here in Kansas City, their input is not a treatment facility, it's runoff from yards, from bird waste, even dog waste if they don't pick it up at a park. That's where we're getting those nutrients, not so much from wastewater treatment plant, but definitely from phosphorus fertilizers.
Thank you. There's a lot of love being shared your way, Laura. There's a lot of people out there love you. And let me see. I think to date, I was trying to count the number of nutrient and HABs projects you and Regina have been on, and I think it's up to 17. So you're
Wow, that's
definitely a
crazy.
rock star.
I've had a lot of help.
Next question is, did you all look at relationships between surface water temperature, water quality parameters, and the respective algal blooms? And then
Yep.
there's a part B, and I'll ask the part B after this.
Okay, so yeah, we did look at that. I didn't present a lot of that data because I wanted to talk more about what the paper presented, but we've talked about this before. In this particular instance, I did a heat map of the 3 different areas and the I think it was probably cell counts from the flow cam, and there was not a correlation to temperature, which I thought was pretty weird. So it's got to be a combination of temperature and maybe sunlight plus the nutrients. Plus, maybe there's some chemical, primer in there that's causing it to go, or some other bacteria that's competing with it. But in this case, we did not have a straight correlation between temperature and cell count.
Thank you. The second part is, and I think if I butcher the acronym, did you all test for microcystin, MCY, A through
Yes.
G genes?
Yep. And I don't know which ones they did at ORD, but that is the part that we really want to look into deeper. We want to do that heat map kind of thing that they did in the paper using the microcystin toxin, and then look at the species that are doing that microcystin production. So I'm hoping that we can, as I was showing some of the stuff I was going to present to Heath and Jingrang, we kind of talked about, we need to dig into this data a little bit more and see if we can pull some more of the stuff out. With 200 parameters, and I don't even remember how many samples we ran, hundreds of samples, there is a ton of data out there, and there's still a ton that we have not really looked through.
Next question. Do these toxins impact dogs, people through dermal impact or ingestion?
So for pets, it's mostly ingestion because they lick their fur after they're in the water, or they chomp on stuff that's in the water, or they drink the water, right? For humans, it's more of a dermal thing unless you're in a big reservoir water skiing, right? So then you could get some contact wreck, you could get some ingestion coming from that way. But certainly in these urban lakes, it's more of a dermal contact, unless it's aerosolizing. A lot of these lakes have these little fountains in them, right? And there's still a lot of study to be done, needs to be done on aerosolization of these toxins and if we're getting exposed that way.
Next question is, what type of sampling done? Surface, any depth profiling done?
Not on this, in this study for sure. It's all surface water grabs. We didn't have a boat out there to go into the lakes. So it was all surface water. We've done some profiling in other studies, but this particular lake was, or this particular study was just surface water.
Okay. Thanks for sharing this work. Can you drop a link to the manuscript in the chat? And I can do that definitely, Troy. It's also actually, I'll put a plug in right now, but I'll say it later. Laura's paper and the link to the manuscript is actually in our Region 7 Science Program webpage, and the link is posted here in this slide.
Yeah, not my paper either. It was totally someone else's paper. I was just a co-author on the little line, but we collected all the samples.
You did a lot of work on that, Laura. You deserve that co-authorship. Next comment. The high-level phosphorus is not only from fertilizers, but from the land and rocks in the lakes.
True.
Another.
Yeah.
Did you want to comment on that further before I move on to the next question, or just?
No, that's okay.
Okay. All right. Holly has she's got my heart. So much mention of dogs, but what about the poor wildlife? Just had to say it.
Yeah, and we don't know how some of this wildlife is affected by the toxins, right? I don't think anybody's looked at that in depth. We've had, cattle die because of their drinking infected waters. But, if you find a dead deer, does anybody go in and do necroscopy, right? Is that what it's called when you do an autopsy on an animal?
The necropsies.
Yeah, so I don't know. There could be other animals out there that are affected by these things. We don't look at it because it's not a human, right? So
Matt, I think you raised your hand. Your question might be related to what Laura just said, so go ahead and open up your line.
Yeah, this is Matt Carlin from the Department of Conservation, and we do have a wildlife health group that is usually swamped with CWD issues, but we do take reports of dead deer and other animals around the state. And we will collect carcasses that are fresh enough and test them if we can get to them in time. And deer in particular, we track because they die near water, but that's usually related to hemorrhagic diseases in the fall. But we do test their tissues to confirm that, and to some degree to disprove cyanotoxins of various sorts. We don't think we've ever really detected a death of a wild animal by a cyanotoxin. We don't hear about every animal, right? And then when we do, it's not fresh enough to test, and we don't send them all. So there's that sort of background screening level issues. But it seems that when animals are dying from cyanotoxins, they're the ones that are pinned into a pond that has an algal bloom and they can't drink it anywhere but there. So I think in general, mammalian and other wildlife that's terrestrial can go drink somewhere else. And I think they understand that. A different sort of question is whether these lakes with repeated cyanotoxin blooms are having a depressing effect on the fisheries in them or not. That's something that I think Dr. Jones at MU has looked into a little bit, the characteristics of lakes that have both cyanotoxins and other kinds of nutrient problems, and then the effects on lake fisheries. But no, that's a great question, and we're always definitely on the lookout for dead animals near water bodies to see what's going on, because there's a number of things that could be different kinds of poisons, cyanotoxin, and EHD outbreaks. So we do screen for that, but don't see much of it.
Thank you. That's interesting.
Thank you, Matt. Let's see here. One more question. Have you seen any testing on the prevalence of HABs before and after the implementation of floating wetlands?
I haven't seen the data. I know that South Lake has installed a lot of floating wetlands out there on their lake, so it'll be interesting to see if that lake changes, because I think those were just installed. I know they had one out there forever, but I think they've just really put more in this last couple of years. So it'd be interesting to see if that has decreased the nutrient levels in the lakes and if that helps. I don't know yet.
Thank you. Marcy, another one of your co-authors and partners in crime, she posted, let me see, it's too small. Marcy, do you want to talk about the graph or chart that you posted in the chat?
Oh, sure. It was just responsive to the question someone had posted on temperatures and how they would relate to the cell counts and the blooms. And so I think if you double click on that, it just shows like the median or I think it's the maximum temperature throughout the study. And as Laura mentioned, they did not correlate with cell counts and not necessarily with the toxins either.
Thank you, Marcy.
Thanks, Marcy.
Next one, shoreline aquatic plant restoration and floating. So this is a response to the wetlands. Shoreline aquatic plant restoration and floating wetland islands can help with phosphorus sequestration.
And as I showed in one of my graphs, right, phosphorus is a big trigger for these organisms. So I'm hoping that that does help a lot.
Another comment, excellent work. Microcystin G is the more toxic and prevalent one. Let's see here. Would the natural filling in of lakes and ponds be adding to shallowing of these bodies of water and combined with the increased air temperatures combined to cause more of these occurrences?
Well, certainly if the lakes are getting shallower with sedimentation, that's going to increase the amount of blooms that are happening, because that you're going to have sunlight into a deeper portion of the lake, so you're going to have growth with that, that photosynthesis. You're going to have more growth in the lake, so it certainly could be contributing. Plus, most of those sediments that are coming in have phosphorus attached to them, so you're increasing the phosphorus load too.
A lot of concern for dogs and wildlife. How much would a dog have to ingest in order to be poisoned?
I'd have to refer you to the Oregon paper. Perhaps I can find a link to that, where they talk about how much dogs ingest and the limits that they've set up for some of the animals. I know that it's way lower than the limits that we have, because we don't drink that water, or after we get in the water, we don't lick our skin off, right? And that's where the dogs are really picking it up. So I don't know the answer to that question, but I would refer you to Oregon. I think it's Department of Health has a really great paper on that.
Thank you. Let's see here. Looks like there's 3 comments related to each other. How does it affect fish and other aquatic wildlife? Someone responded and said oxidative stress, impaired growth and development, disruption of the immune system, gill damage, liver damage, and then it says we saw dead turtles and fowl, but we don't know for sure how they died. Do you have any comments on that, Laura?
Nope. I haven't looked at fish, to be honest, or any other aquatic organisms. I do know that sometimes they affect them and sometimes they don't, probably dependent upon what they eat. I did just, Chris Nietzsche just gave a presentation on some work they're doing in their mesocosm, and they were seeing definite effects downstream from a toxic bloom that are affecting the macroinvertebrates, the bugs in the water. So that's the beginning of the food chain there, right? Those bugs are eating the cyanobacteria, and they are changing. So I don't know the exact answer to that, but I'm sure there are effects.
Next one is people are putting these into perspective, Laura. So someone said, my mom's neighborhood, Big 11, Kansas City, Kansas, down the street from the old regional office.
That's it.
Mm-hmm. Let's see, Mark Lesher says, we need to educate surrounding landowners and residents to limit their fertilizer use around these lakes. Are there any outreach efforts to make people aware of some of the actions and the consequences?
I think South Lake, I know they have active outreach surrounding that lake. I don't know, Marcy, if there's stuff going on in Region 8 in that regards, but certainly something that we need to take to those neighborhoods, perhaps as part of our environmental justice outreach here in the Kansas City area, that could be part of that process.
Lots of thank yous for the presentation. The last question is, what's the better treatment, copper sulfate or a solar-powered traveling fountain?
I don't like copper sulfate because it has a tendency to affect a lot of organisms in the lake, not just the cyanobacteria. So I would never really tell somebody to go treat with copper sulfate. We actually saw an instance in one of our earlier studies where the blooms were kind of moderate before they treated with copper, and afterwards, boom, they just blew up after they recovered from the copper sulfate treatment. So peroxide has been used often. I know KDHE has done several studies with the peroxide treatment. That seems to be the way people are moving, I think, to treat these cyanobacteria. Fountains are great. They do oxygenate, they ozonate the water. My only concern with that is like I said, the aerosolization. Are we breathing that in as we're walking around that lake? I don't know.
Someone shared a link to the Kansas City. There's a regional water quality education committee. Thank you for that.
That's cool.
And again, some more thank yous. Well, thank you, everyone. I want to thank Laura and for taking time away from your work to give this webinar, and for everyone attending this webinar. Thank you so much. The recording to this webinar will be posted in 2 locations, the Region 7 Science Program SharePoint site, which is internal, and the Region 7 Science Programs webpage, which is external. Our webpage is a one-stop shop for Region 7's research projects and ORD's funding opportunities, webinars, trainings, and publications. As I mentioned earlier, there is a link on our webpage to Laura's work and the publication. You can click on the link to our webpage and hover your phone over the QR code, and it will take you there directly. Our Region 7 Science Program webpage is updated monthly, and we post all the STAR, P3, and all other ORD research funding opportunities. And if you're interested in learning more about the research we do in Region 7 and more projects that Laura and Regina have completed in HABs and nutrients, feel free to check out our story map. That's an external website as well. I posted all the links in the chat at the beginning. I will put another one right now at the end. 1 is internal and 2 are available for external facing. If you have any additional questions for Laura, feel free to reach out to her. And if you have any questions about the Region 7 Science Program, feel free to reach out to me. Thank you all for attending and have a wonderful day.